Thank you for the opportunity to present here. It's a real pleasure to be here to talk to you about the role that LifeArc plays in establishing strategic partnerships to translate the innovative scientific discoveries all the way to the clinic. So I'll start off by giving you a very brief introduction to LifeArc and our track record. You heard a little bit about LifeArc and what we do from my colleague Veronica in the panel. I'd like to go into more detail to discuss our partnering business model, showcase some of our antibody platform capabilities, and then really finish off with some partnership spotlights. So as Veronica mentioned, LifeArc is a self-funded medical research charity with a focus previously on early stage translation, but we're now also moving to late stage translation and bridging the gap or the valley of death, as we've already heard about, between the innovative scientific discoveries into medical breakthrough for patients in areas of unmet medical need. So by collaborating with researchers and universities and industry partners, we aim to accelerate the development of new therapies that can really improve patient outcomes. So we have an excellent track record in and expertise in humanizing antibodies that continue to represent life-changing treatments to patients globally. So over the 30 years, we've humanized 95 antibodies with a success rate of 98%, and 5 of which are on the market. We've worked with 51 companies worldwide with a number of repeated business partners, including world-leading academic centers in the USA. So we're extremely proud of the contributions to the development of these drugs that are currently on the market. Our earliest success stories were Entyvio, Actemra, and Tysabri, and the most recent Keytruda, which we all know is one of the world's top-selling drugs, and also lecanemab, which was approved last year. We're also very proud of the fact that for the case of Keytruda and lecanemab, the sequences that actually left our lab many years ago is the actual sequences that's being injected into patients today. So although the molecules changed hands of very many companies, the sequence remained the same, which is a true testament to the expertise of our team. So many of you may be familiar with the story of Keytruda and how it was brought to the market. But for those of you who may not be aware, I just thought I'd put up this slide. So our humanization activities were really based upon Sir Greg Winter's patented technology based on CDR grafting, which we continue to use today. So Organon actually approached LifeArc, or MRC technology as we were known then, to humanize their lead candidate molecule, which was called PD109A at the time. So they then went on to file the patent, and then that was then licensed to Merck, approved in 2010. The FDA approved in 2014, and is one of the best-selling cancer drugs today. So it's really due to the financial sustainability of the situation that we're in, because we're self-funded from the royalties that we get from Keytruda, that we can actually partner with innovators across the world, wherever you're based. If you're looking for that truly collaborative partnership, as you heard from the panel, we believe that, working for LifeArc, we can really deliver that. So, as I mentioned, we've worked with many different partners across different companies. So we work with academics, research institutes, startups, and also biotech companies. So as Veronica alluded to, we see ourselves as enablers in the ecosystem to really remove the common barriers that they face when translating their innovations. So as Veronica also mentioned, we have a unique partnership model, and we are truly driven by patient impact. So for antibody discovery projects that enter our pipeline, you know, they should have strong therapeutic rationale, target-specific and a disease-relevant mechanistic data. And we also appreciate that you can't have one partnership model that fits everybody. So we're really flexible. We can, have bespoke partnerships to really tailor the needs of our partners, and also dependent on the scale of work. For antibody humanization, we rely on our partners to come to us with the parent antibody and the target that is validated. So we ask our partners to have a very strong data package. Again, we can do both of these projects at no R&D cost to the partners. They will also retain the IP and the commercialization control. I should also mention that that's not where our relationship ends. Once we hand over the lead candidate, we can also provide post-project guidance, including patent support. So what do you get from partnering with us? So in addition to our expertise and technologies, as I mentioned, we really offer a unique business model to our partners. So we can progress your project at no cost, whether that's antibody humanization or discovery. We take on the financial risk of funding the R&D cost. We appreciate that a lot of academics or early stage biotechs may not have the money to do this, and if they are in a fortunate position to have the funds to do this, we can fund this so they can then use those funds to carry on the development work. Importantly, you retain your IP, and then we have a very collaborative approach, and we consider any target, modality, and therapeutic indication. So now I'll go on to talk to you about our discovery and humanization platform. So our biologics discovery and development team is based at 2 different sites. Most of our R&D scientists are based in the Stevenage site on the GSK campus, and we also have a small team that's based at the Francis Crick Institute in London, where we house our early stages of antibody discovery. So the team is split into 4 different main areas. So we have an antibody humanization team, an antibody discovery team, and then we have a screening and lead characterization team that support all of our antibody humanization and discovery programs. In addition to that, we also have a protein and analytical sciences team that actually support the antibody humanization and the antibody discovery teams, as well as the screening and lead characterization teams. So this slide really just summarizes our capabilities. So we see ourselves as that one-stop shop for antibody discovery and humanization. So I mentioned earlier about our antibody humanization capabilities, but in 2019 we in-licensed a fully human transgenic mouse platform from Kymab. So the Kymouse platform, they were obviously based here at the Babraham Institute. So we're really in a fortunate position to use innovative B-cell selection technologies and the Kymouse platform to generate fully human antibodies. So our partners would come to us with a validated target. We have the ability to generate the immunogen and screening reagents. We adopt multiple immunization strategies to ensure that we can deliver you the right candidate molecule that fits your target product profile, and then go on to do single B-cell sorting and clonal selection. For antibody humanization, the first step is for us to generate a chimeric antibody, which will serve as a positive control for all of the downstream processes. Our team will generate a library of of humanized variants, and we have no limit into the number of humanized variants that we will generate, and we will mix and match every heavy chain with every light chain variant to produce a library of variants, and then the output of both the discovery and humanization programs then go through a high throughput screening method to then actually generate a lead candidate panel We have in-house protein production facilities, so we generate all of our antibodies in-house using a two-step process, affinity purification as well as size exclusion. We also have antibody engineering capabilities, so if we need to engineer out any sequence liabilities, we can also do that. We have a molecular and cellular pharmacology team based at Stevenage who support all of the functional potency assays as well. And then towards the end of the program, we do a detailed developability assessment to ensure that what we deliver to our partners can actually be manufactured. So the aim for both of these programs is to deliver to our partners a lead candidate antibody plus backups that are then optimized for clinical testing. So this is a summary of our antibody discovery platform, which will be no surprise to many. So we've called this our B-Smart platform, which stands for B-cell screening method for antibody therapeutics. So as I mentioned, immunization is absolutely key. So we adopt multiple immunization strategies. So we can do immunization using proteins, peptides, DNA, cells, and also messenger RNA lipid nanoparticles. And then we have two innovative single-cell B-cell screening technology. So we use the FACS as Kymab did, but we've recently also bought the Beacon technology as well, which you can screen using function first and also use PBMC. So we can really work with clinicians as well to identify antibodies based on function. So again, the Beacon technology was heavily utilized during the COVID pandemic. We then use single-cell sequencing. So again, this offers many advantages. You've actually got the paired heavy and light chain sequences of your antibodies coming from a single cell. And then we've got high throughput production and screening. So we can screen, more than 100 candidates in a 384-well or a 96-well plate to generate a lead panel to characterize further to deliver 1 to 3 candidates to our partners. For antibody humanization, the first step, as I mentioned, is a feasibility stage where we generate the chimeric antibody, and we will also confirm the binding and confirm the data package that has been sent to us by our partners. We then go on to do the humanization sequence design, so we still use a combination of CDR grafting and molecular modeling. We will also do some sequence analysis, and again, we can not just do humanization of classical IgGs. We've worked with single domain antibodies, heavy chain only, fused to FCs, as well as VHH. Again, we've humanized antibodies that have been generated in mice, rabbits, chickens, so we have a lot of experience there. And then, as I mentioned already, we do high throughput expression and screening. And then with our lead candidate panel, we again repeat all of the assays and do a full assessment of the binding, do a host of biophysical assays, and then also test the humanized variants in a functionally relevant cell-based assay. So in terms of high throughput screening, all of our antibodies are purified in-house in a 384-well plate. They're all quantified and then normalized. We can also do high throughput antibody purification and QC. We do a detailed kinetics. We have the BioCore 8K that can help us do that. And then we've recently purchased the Cartera to help us do the epitope binning. We also do cross-species selectivity, so make sure that your antibody is going to bind to the mouse and human target, as well as cyno for downstream development assays. Again, binding to the cellular target is also key. And then our molecular and cellular pharmacology teams can also support a number of assays, such as internalization assays for ADC targets, as well as ADC-CC, ADCP, and CDC reporter assays as well. We also, as I mentioned previously, we do a host of biophysical characterization assays to really increase your drug's chance of progressing to the clinic. So in addition to looking at the levels of purity and aggregation, We do solubility assessments. We look at non-specific protein-protein interactions. We do a host of accelerated stability assays. We look at the melting temperature and the aggregation temperature. Towards the end of the program, we also do serum stability. So we look at the ability of the stability of your antibodies in the presence of mouse, human, and cyno serum, and also look for non-specific interactions. So I'd just like to share some specific partnership spotlight. So one of the key things that I'd like to highlight here is, a case study that we did working very closely with a company called Keynote Biotechnology, where we humanize their VHH targeting TROP2. And, TROP2 has been, a very promising target, but actually what we've humanized for them, they are going to take that forward as a naked antibody. So it's a first-in-class naked antibody. And we humanize that, and within 2 years of the humanization, they've actually formed a strategic partnership with CRH to take this forward into clinical trials. So really showcasing the importance of working with the right partners and the expertise that LifeArc has to actually progress these innovative ideas to the clinic in the shortest time possible. And this is just really a testimonial from Gordon at Kisoji. So, even though we are working, in different areas, it's really important, that you pick the right partner who can really bring each other's expertise to drive that patient impact. Sorry, it's taking a long time to get to the next slide. And another case study that I'd like to also showcase is that in collaboration with... Have I got 1 minute? Okay, I'll be quick. Is a collaboration with Andrew Mackenzie at the MRC, LMB in Cambridge. So together we generated an IL-17 antibody, which we then went to humanize. And this was then partnered with Sinomab. This is now in clinical trials. And just earlier this year, we've had positive phase 1b results for the treatment of moderate to severe atopic dermatitis. And then just lastly, we've had a track record between LifeArc and Cleveland Clinic. We've worked together on a number of humanization programs. Cleveland Clinic have actually even created a spin-out company focusing on the nanobody that we have humanized. So it really just shows the true value and how collaborative partnerships such as these can lead to quick value creation, translation, and patient impact. And just earlier this year, we signed a master service agreement for the development of new monoclonal antibody therapeutics with Cleveland Clinic. And finally, I hope you've had a chance to speak to my colleagues, Veronica Gray and Lucy Buckingham. If you haven't had a chance, we have a stand outside, and we'd be happy to talk to you during the networking session. So with that, I'd like to thank you for your time and happy to take any questions during the networking sessions.